Want to thank everybody for coming. I think people are still starting to trickle in. So I just wanted to introduce and welcome Paul Valdmanis, um, and tell you a little bit about him. So he comes from the north. He started his undergrad at University of Ottawa and does PhD at McGill before coming and doing his postdoc work at Stanford. And since 2017, he's been at UW with the Department of Medicine, Division of Medical Genetics, where he is now an associate professor. So he's done a cool variety of different topics from ALS and neurodegenerative diseases, including some AAV gene therapy and a lot of work on molecular mechanisms and genetic mechanisms of disease. And one of the interesting things here, there's a TDP-43 paper was one of your first papers and one of your most recent papers. So there's definitely some through mechanisms and pathways that you've been working on through your whole career. So I'm really excited to hear what you have to say and hear what your topics, hear what your research is on illuminating the dynamics of tandem repeat expansions. Yeah, I'm looking forward to hearing what you say. And people can ask questions either in the chat or raise their hands, I believe, afterwards, and we can moderate those when we get there to the end. I'll pass it over to you then.
Super. Thanks so much, Michael, for the introduction and the invitation to speak here today. As Michael mentioned, my lab is interested in neurodegenerative diseases, and I have a long history of trying to understand novel genetic risk factors for amyotrophic lateral sclerosis and Alzheimer's disease, especially using long-read sequencing approaches. And I'll talk a little bit about that today. So we're really interested in, various pathogenic mechanisms of disease, and I'm sure everybody's familiar with, types of pathogenic variants, single nucleotide variants, deletions, insertions, and copy number variants. But what I'm going to talk about today are these tandem repeat expansions that are these groups of, like say 3 or 6 nucleotides that are copied over and over again in the genome of individuals that when they expand to a certain length can lead to disease. And not just any disease, but over 50 tandem repeat expansions have been known to lead to both neurological and muscular disorders. And these tandem repeats can be divided into 2 different components. They're short tandem repeats. These are ones with a repeat motif of 2 to 6 base pairs in length. So you can think of like a CAG repeat expansion in the Huntington gene, in the first exon of this gene. So CAG repeated over and over again will predispose to Huntington's disease. But you can have other repeats in the 5' UTR, introns, exons, 3' UTR, and several of these different flavors of repeat expansions are going to cause disorders, including hexanucleotide repeat. That's the most common form of amyotrophic lateral sclerosis. And these are the short tandem repeats. There's also something called variable number tandem repeats, and these are much longer internal sequence. By definition, these are the internal sequence of each repeat is more than 6 base pairs. But from a practical standpoint, these are about 20 to 80 base pairs in length. These chunks of 50 base pairs repeated over and over again, this exact same sequence. So these are the VNTRs. These are the larger kind of sequences. Repeats can have they can help predict, not only disease severity, but also age of onset of disease. And so a classic example, again, is the CAG repeat expansion in the Huntington gene. And depending on the number of repeats in an individual, if you have fewer than 36 repeats, you're really not likely to develop Huntington's disease. But as you can see, each additional copy of the repeat decreases your age of onset, such that if you have 39 copies, your predicted onset is just before the age of 70. But individuals with 50 or more copies, will have a predicted onset 40 years earlier than that. And there are sequence interruptions within this repeat motif that can alter this risk and the age of onset. But this kind of indicates that often it's the CAG RNA repeat rather than the polyglutamine protein that is critical for this age of onset association. And if you want to learn more about Huntington's disease, Tom Bird in the Division of Medical Genetics in the Department of Neurology has written an excellent book on the topic of individuals that he's seen with the CAG repeat expansions in Huntington's disease. Now, some of these repeat expansions, and even some of the VNTRs, the larger repeats, are not necessarily bad for you. Some of them are beneficial or um, innocuous in terms of phenotypic presentation. This is some examples of a tent 5A VNTR length, and this is different lengths of this 15 amino acid or 45 base pair coding repeat, and each additional copy of this repeat adds another, zero point 0.3 centimeters to your relative height. So the longer the repeat, the taller the individual. And Poe Rouleau at Harvard and Ronan Mukamel and have demonstrated other examples of these associations between repeat length and phenotypic traits. These repeats are not just in mammalian genomes. I think one of my favorite examples of repeats that have evolved to develop beneficial functions comes from bacterial space, where there are three different genes that decorate the cell matrix with different kind of moieties, GalNAc and phosphatidylcholine, expressing moieties on the top of in front of the cell that help in cell recognition, attachment, and cell entry. And there's actually tetranucleotide repeats within the coding region of these genes, and the bacteria recognize that the mutation rate of some of these tandem repeats is orders of the magnitude greater than code base pair changes in the genome, and such that if these tandem repeats mutate to different length, some of these by chance are going to lead to premature termination codon and prevent expression of these proteins. And if these happen in the right combination of genes, you can actually completely remove the cell moieties that are present. And so you can imagine if these are important for cell entry, having certain moieties of the surface can help in entry and attachment into cells, but they're also help promote immune response and targeting from the immune factors. And so if you kind of have this transition between presentation of these moieties and removal, you can kind of like shapeshift and evade some of the immune responses, lay low for a while in quiescent phase until kind of the immune response has passed, and then re-establish some of these based on some of the changes in the tandem repeat length. Anyway, so this is kind of a cool kind of examples of some of these repeats, but kind of a long-standing question that we've had in the lab is, really how do these repeats expand? How do you go from, a handful of copies, 6 or 8 copies of the CAG to, 40, 60, or even 100 repeats in Huntington's disease? And what is the process involved in this expansion? Is this a stepwise event? Is this kind of a catastrophic event that massively leads to this expansion? And where do these repeats add? Is it the beginning, middle, or end? And really, for a long time, there wasn't a very good system for identifying, exactly this internal sequence. And this is an example of this hexanucleotide repeat in C9orf72, which was identified primarily through classical approaches like Southern blots, where radio-labeled probes can, after digestion of DNA, reveal some of these larger repeat expansions in individuals with ALS. And these repeat expansions could be confirmed with PCR with labeled probes that can indicate this kind of like a hairpin-like, the stepwise kind of process that indicates that these individuals here have larger repeats, but it doesn't tell you exactly the internal sequence of the repeat or exactly how many copies there are. There have been a lot of advents in long-read sequencing approaches to kind of circumvent some of these approaches to be able to identify the exact length and internal sequence of some of these repeats. Two that we use in the lab include PacBio single molecule real-time sequencing that adds these kind of smart bell adapters after digestion of DNA, reads sequences over and over again, and with enough passes, after each iteration, it can resolve the actual sequence of these larger segments in kilobases in length with fairly high accuracy, approaching kind of the accuracy of Illumina short read sequencing. And then other technologies, including Oxford nanopore sequencing, use pore-based approaches to identify the actual sequence of a long DNA molecule that can be megabases in length that really can help piece together larger structural variation within the genome. And these approaches have really, again, revealed surprising insight into some of these really large expansions. One of my favorite examples is a pentanucleotide repeat in the SCA10 gene, leading to spinal cerebral ataxia. And this is a repeat that is expanded, especially in South America, up to 1,000 repeat copies of this five base pair repeat motif. But individuals with this repeat, are largely unaffected if they have a particular repeat motif, this ATTCT. But individuals shaded black here, where there's just a one nucleotide shift from ATTCT to ATTCC in teal, are the ones that suddenly develop this motor neuron disorder, spinal cerebral ataxia, that it impairs kind of walking and mobility. And you can see that, individuals that are phenotypically normal start out with this gray motif, and then halfway through the repeat, it switches to this teal motif, and that's what is leading to this disorder in this family. Kind of underscoring the necessity of identifying both length and internal sequence of the repeat. So as Michael mentioned, we're really interested in amyotrophic lateral sclerosis. This is a disorder also known as Lou Gehrig's disease that is caused by the degeneration of motor neurons in the brainstem, spinal cord, and motor cortex. The onset of individuals with ALS is in the 40s and 50s, and really rapid progression to death within a few years. And current drugs can only prevent progression by about a month or so. There's about 5 to 10% of individuals with family history, and those individuals have helped lead to some of the identifications of some of the key genes in ALS. And we've been involved in some of this process, including pathogenic variants in TAR-DBP, which is also known as TDP43, and FUS, kind of in the late 2000s. And then C9orf72 repeat expansions that can cause up to 50% of familial cases and up to 10% of individuals without a family history of disease. But even with all of these genetic studies, there's still a fair amount of missing heritability. There's a lot of cases where the genetics can't account for, the susceptibility to ALS alone. And so we hypothesize that long-read sequencing and kind of next-generation sequencing approaches could fill maybe some of this missing gap. And really help explain another conundrum in ALS, where there are, again, TDP43 and FUS, which are both RNA binding proteins, don't seem to associate with C9orf72 repeat too much, which is a repeat expansion. So it suggests that, perhaps there are other RNA targets of these RNA binding proteins in ALS. And we've worked a lot on sequencing in families with ALS, including multi-generation pedigrees. And part of this sequencing led to this really unique variable number tandem repeat with a 69 base pair internal sequence copy in the last intron of a gene called WDR7. And the reference genome has about 6 copies of this repeat. And you can see that it folds into this really strong stem loop-like structure where nucleotides on this side base pair with ones on the other side, forming this really tight hairpin repeat motif. And so this is the reference copy genome with 6 copies of the repeat. And then in the familial case with ALS, you actually have 37 copies of this repeat. What's more intriguing is that each copy of this repeat actually has a predicted binding site for both TDP43 and FUS, which binds to the exposed loop of a stem loop structure. And so you can imagine that if you have more copies of this repeat, the more likely you're going to have association with some of these proteins that are aggregating in disease. So we wanted to see what kind of the length of this repeat was in a large cohort of individuals with sporadic ALS. And we were fortunate enough to be able to use kind of classic approaches like PCR, where we could design primers before and after the repeat. And this is kind of an example with 4 repeat copies. But you can see, each of the individuals that we were sequencing often had 2 repeat copies, one from the mother and one from the father. And we could score these and count these across a large cohort of individuals with ALS, as well as controls. These were from populations from Answer ALS, as well as from Quebec, Canada. And we can see that individuals with ALS were biased towards this kind of larger repeat copy, including one individual that had 86 copies of this repeat. This is a woman with sporadic ALS that developed it at the age of 72. But of course, this is just again the length of the repeat. We really wanted to know what the internal sequence was in case there was some kind of specific sequence that helped in this association with some of the RNA binding proteins like TDP43 and FUS. So what we did was we designed actually appended to the PCR primers for this repeat 12 barcodes before and 12 barcodes after to allow us to kind of multiplex across this repeat in 100 individuals. And so this was great. We were able to get the sequence, but we kind of came across a problem is that, this is what the 86 copy repeat looks like, is you have these repeats over and over again in the sequence. And how are we going to be able to identify, whether there was a particular pattern associated with disease? So we started breaking this down. We started noticing that, on top, this is the canonical 69 base pair repeat motif. And there were a lot of actually internal sequence differences within this. And these sequence differences weren't random. They were occurring at 6 places throughout that 69 base pair repeat, or sometimes there was a 1 base pair deletion. And so one of the things we started to do was to color code each of the individual repeat motifs, essentially, to try to see whether there was a pattern. So to do this for each individual, we would start to identify the start of the repeat motif, kind of left justify these and identify kind of based on the color coding what the different repeat motifs were. We called each of these a single amino acid letter to be able to use protein sequence alignment programs to help align a lot of these large repeat copies. And we color coded these as well. So for instance, at the top of this repeat, you had C and G changes, and we would call this letter H and color it green. In the second kind of row, we would call it E. It had these two A's that were different than the canonical sequence, and this would be purple. And then the third repeat motif was the same as the first. It was green again. Fourth was purple. And then the fifth had another nucleotide change here. Okay. And so when we did this, we were then able to kind of color code and align the entire set of 288 alleles across the x-axis, and then align the repeat length on the y-axis. So you can see here, for instance, the first kind of handful of individuals, these are people that had 4 repeat copies, 3, 2, and 5. And if we did this across all of the 144 individuals, we started seeing these really striking patterns. And we could start to see clusters, whereas, for instance, a family with ALS would have certain repeat motifs, ALS versus control, and Parkinson's disease as a non-neurological control. But what we started to see was that there really were these kind of differences in repeat length from, 1 copy up to 55 repeat copies. And we also started seeing patterns of clusters where they seemed to kind of be grouping together based on commonality of sequence motif. So even though the length might be very similar, the internal sequence was quite different among these groups. And this is very similar to, the plot looked a lot like a Manhattan plot for genome-wide association studies, but since we're in Seattle, we called this the Seattle plot and overlaid the Seattle skyline with some of these peaks that we were seeing. But we could really start to identify now certain patterns associated with these repeat expansions. So for instance, we would rarely see the same repeat motif side by side. They were often these kind of appearing 2, 4, 6, 8, so on, repeat motifs apart. So these seem to be really expanding in groups of 2 repeat motifs instead of 1 itself. So instead of 169 base pairs, there's 138. We started seeing individuals with these kind of larger duplication events occurring within the same repeat motif, as well as you can see that there's a very strong periodicity, not only periodicity, but directionality of the repeat. So there was a long stability at the 3' end, and then a lot of variability at the 5' end. And anytime we saw a private or unique repeat motif with nucleotide changes not observed in anybody else, these were always appearing at the 5' end of the repeat motif, suggesting this is kind of a dynamic expansion that is growing in a directional manner from 3' to 5'. This directionality was even more underscored when we would kind of break apart populations of some of the samples that were sequenced, and we'd use some of the 1,000 Genomes Project samples for this analysis. So you can see here, if you group individuals by their superpopulation, individuals of East Asian ancestry and American mixed American ancestry were very similar as this repeat was expanding throughout human history until you got to this break point, perhaps, where there was separation between the two populations. So you can see everybody in the Americas started had this pink allele here versus other iterations in East Asia. We saw other breakpoints as well that would separate, for instance, samples of African ancestry and East Asian ancestry that are denoted by the arrows here. Again, revealing kind of fascinating insights of how some of these repeats are expanding across human evolution. And then from a human genetics perspective, I was very fascinated to be able to track, versions of these repeats across multiple generations in a single pedigree of individuals with ALS. And then finally, I haven't really gotten into kind of the function of WDR7 and the protein that harbors this repeat. We're currently working on a mouse model of this disease to try to see, what the phenotypic presentations are. But at least it makes sense in terms of where this protein is expressed at the synaptic membrane, involved in RAB3 recycling and synaptic vesicle recycling. So we think that this, when this expansion occurs, it can impact the ability to appropriately recycle the synaptic vesicles in neurons. Okay, so this work was published, but it really kind of inspired us to take a look at repeat expansions more broadly throughout the genome. And work from the Eichler group has profiled, some of the places where both short tandem repeats in blue are present throughout the genome, and actually the frequency of some of these variable number tandem repeats is considerably larger, especially you can see here at telomeric regions or subtelomeric regions. There's this real pileup of some of these VNTRs. And there's over 400 of these that are unique to humans that are present only in one place in the genome, allowing us to kind of precisely map out using some of the same principles we use for WDR7 for other tandem repeats. And we really wanted to kind of get a sense of the lay of the land. And for this, we took advantage of two large cohorts of individuals that have undergone phased long-read sequencing, including from the Human Pangenome Reference Consortium, the HPRC, as well as the Human Genome Structural Variation Consortium. And these, again, are performing long-read sequencing of samples from the 5 superpopulations available from the 1,000 Genomes Project, from American populations, African, South Asian, East Asian, and European. And with all these populations, we can start to get profiles of, what some of these tandem repeats look like. And so again, we're making these Seattle plot-like profiles of some of the individual alleles from some of these long-read sequences, and trying to see, okay, for instance, this is a 41 base pair VNTR in a gene called NWD2, not necessarily associated with disease, but you can see that there's a fair bit of variability in repeat length from individuals that have 3 repeat copies up to 45. And we could, again, color code these, and you can see that most individuals have this blue color, this blue motif, but sometimes this will switch to this purple, this 1 base pair change. And then from purple, it'll switch to green, where there's another change here. So for the most part, a lot of the VNTRs that we kind of would profile this way had this type of, differences in repeat length, but commonality in a lot of the repeat motifs. We found other cases where every single repeat motif was a different color, meaning that the internal sequence of each copy of this repeat was different. But with this, we could also see in instances here in white where there were gaps. So there were deletions in the repeat motif, and then I've boxed here other places where there's actual duplications in certain individuals, and you can see based on the coloring of these combinations where exactly the breakpoints of some of these expansions and contractions are taking place. So we can start to kind of build together a model of how these repeats are expanding and contracting. Other repeats looked a lot more like WDR7, where there wasn't a very strong, consistent pattern among individuals, and instead had these different kind of subclasses of repeat expansions. In this case, a lot of the expansions seem to be occurring in the internal to the repeat itself. And when, again, we would overlay the population of origin of some of these samples, we actually saw that there was a real kind of differences in repeat frequency, internal sequence frequency, based on the population of samples. So some of the repeat motifs were present almost exclusively in samples of African ancestry, whereas other ones were not present at all, and almost exclusively in populations out of Africa. And so we could use this again to demonstrate that certain repeats seem to be expanding and evolving very quickly throughout human history. Some of the repeats had their own really striking features with internally, where there were some sections of the repeat that had different repeat motifs at the beginning, followed by a pure repeat motif, followed again by kind of combinations of three repeat motifs. Here you can see, and this is these are groups of 33 base pair repeat motifs. So this, if we think that these repeats are expanding in groups of 33, this would add up to 99 base pairs. Very similar to the 138 base pair, di repeat that seem to be occurring for WDR7. So we're starting to get a sense that there's a kind of a magic number where some of these repeats expand, and this is consistent with the Okazaki fragment and lagging strand DNA replication, where these fragments 150 base pairs apart may not be appropriately finding their sequence in this repeat expansion, and that is precipitating some of these expansion events that we're observing in these VNTRs. And that would be consistent as well with this idea that for Huntington's disease, when you get to this threshold of about 40 repeats or more, that's when you really start to see a phenotypic presentation. So again, 120 base pair total repeat length. Okay, so this is, again, some of the work that we've done trying to look at population-specific differences in repeat length and composition in modern humans. There are also archaic humans that have introduced DNA into modern humans as well. So obviously, a fun kind of parlor cocktail hour discussion is, what percent of Neanderthal DNA you may have. And Neanderthals and Denisovans are examples of two ancestral hominids that have emerged out of Africa, Neanderthals populating much of Europe, and then Denisovans originally in oceanic populations, especially in Melanesian populations, named after this cave in Siberia where there was a hermit named Denis that lived, and so this whole hominid species is named after one individual's cave there. And so, we've been really interested as well, not only in modern humans, but also in archaic populations, how some of these DNA repeats may have expanded and integrated into modern humans. And our collaborator, Emilia Huerta Sanchez at Brown University, has done a lot of work looking for signatures of positive selection. So areas where Neanderthal or Denisovan DNA has been introduced into modern humans that has conferred a selective benefit. One example that you provided in the gene EPAS1 is shown here, where there's a series of 5 Denisovan tagging SNPs that are present almost exclusively in Tibetan populations that are associated with protection against hypoxia in high altitude environments. And this work in humans is also modeled in horses, where EPAS1 is also a signal for positive selection. And in the bottom, you can see even a coding change, an R144C change, tracks with the altitude at which the horses were sampled. So horses at high altitude in this red line have a much higher alternative A allele frequency or heterozygous allele frequency than horses sampled at low elevation populations. Suggesting this variant is functional and helps in an adaptation to this environment. So Emilia Huerta Sanchez has gone on to identify other regions of the human genome with examples of positive selection or integration from archaic populations, and has kind of lists of regions in the genome, including this region on chromosome 12 that harbors several genes, including LRRK2 and MUC19. MUC19 actually here is one of the top genes when you consider signatures or at least high minor allele frequency in a population specific manner. So kind of the way to read this plot is that these are this is the number of individuals with that have low levels of African-specific alleles that are fixed in Denisovin, so 100% in Denisovin variants, and then 30% or more in a population of interest. And in the Mexican population, MUC19 really shows up as really highly enriched for variants um, that are Denisovin specific, not present in Africans. And you can kind of overlay intergress tracks across populations and find the profile of this kind of region on chromosome 12 near the MUC19 locus, where there's high levels of integration, high levels of Denisovin selectivity. And this is actually kind of almost like a layering of integration. There's actually both Denisovin specific variants at this region that are overlaid onto a Neanderthal background, suggesting that Denisovin variants hopped onto the Neanderthal allele, which then hopped into the human allele and then became highly selected for and a signature for positive selection in American populations, especially in Mexico. So when Emilia Horta Sanchez discussed some of these findings, we immediately became interested because MUC19 is one of the about 21 mucin genes. And these genes are particularly interesting because they all harbor some of the largest coding variable number tandem repeats in the human genome. And so this is kind of a UCSC plot of MUC19. You can see how many exons here in these thick bars here in blue are present before and after this section here. And this is a region, this is one exon here that has a 30 base pair variable number tandem repeat. And the VNTR covers almost the entire exon. This exon is over 10,000 base pairs in length. So again, so if this region has the signatures of positive selection and Denisovin integration, and also harbors this VNTR, we wanted to see, using some of the tools that we've developed in the lab, what is the length of this VNTR? And is there, could this help explain some of these signatures of positive selection? So Eli Kaufman, a bioinformatician in the lab, has developed a program called Inventor, or isolated Neaton VNTRs. And from this, we can collect from the genomic sequence reads, the phased reads. We can cut out sections before and after VNTR and the VNTR sequence, and then align them into those different repeat motifs like I was showing earlier. And from this program, we could actually see that the repeat length for MUC19 is fairly stable. Some individuals have about an 8,000 base pair repeat motif, others with the 10,000 base pair repeat motif. And then in rare instances, you start to see these much larger segments of this VNTR. But in American populations, you had 7 independent individuals with the exact same length expansion at this site that led to a repeat length of almost 23,000 base pairs. And if you break down the repeat length into its individual repeat components, we could start to see patterns of these groupings of kind of like hierarchical expansion events taking place at this region. It's almost like a super VNTR, and you can see kind of color-coded how you can have 5 exact copies of a 3,171 base pair VNTR. And so this is a 30 base pair repeat motif in the coding exon, and however, actually 3 copies of this repeat motif for 27 base pairs instead of 30. And so that's why it is still divisible by 3. It still preserves the reading frame of this exon, but there's not a 0 at the end of this. And so this is kind of a schematic of this massive repeat expansion that we're seeing in the exon 56 of MUC19. Most modern humans have a 3 kb segment in the middle of this 30 base pair tandem repeat, and the reference genome has 287 copies. But this 3 kb segment expands and is copied 5 times in this exon in American samples, and these are flanked by these Denisovin specific variants that are involved in this integration. And what's really neat is that, up until this point, some of the largest coding exons in the human genome include another mucin MUC16 that's 21,000 base pairs in length. Titin is 17,000 base pairs. This expansion actually leapfrogs these to generate kind of one of the largest coding exons in the human genome. And this coding exon is actually involved in the glycosylation of PTS amino acids. So it's a PTS domain that's involved in this mucin. The mucins are these proteins involved in lubricating the lung, the trachea, and other kind of organs, and are particularly involved in the front line of defense against pathogen infections. So the expansion is predicted to increase the size of this PTS domain and kind of double the size of this region in these individuals. What is neat is that now that we have this information, we were interested in going back to kind of a lot of the short read data that's available in biobanks and other kind of cohorts. And we wanted to see, okay, if we have individuals that both have long read and short read data, can we accurately predict the length or estimate the length of this VNTR, even if we don't have long read genomes? And so on the left, this is an example of, for instance, the WDR7 repeat. And these are individual sequence reads on a IGV track. And you can see that, there's the density of the reads is fairly consistent across non-repetitive regions. But as soon as you get to the VNTR, you have this pileup of reads suggesting that the repeat expansion in denisovans at WDR7 is much larger than the reference genome at this position. Conversely, Neanderthals actually have a paucity of reads in this, so the number of reads that map to the VNTR is much lower than the rest of the background genomic region, suggesting that Neanderthals have a shorter repeat for WDR7 compared to the reference genome. And we can do this iteratively across the dozens of samples that we have long read and short read data for, and we can get a fairly accurate estimate for what the repeat length is using some of these calculations and compare that to the two alleles based on from long read sequence data. So with this information, then we can go and try to take a look at what the repeat length was for MUC19 across some of the superpopulations in the 1,000 genome sample. And again, in American populations, you have this tail of individuals with much higher number of repeat copies or length of this repeat. And this is particularly pronounced in samples of Mexican ancestry compared to Colombia, Puerto Rico, and Peru. What's more is I sent this information to Dr. Huerta Sanchez's group, and they overlaid this with the number of individuals that had 1 or 2 copies of Denisovan-like integrous tracks. And you can see that 1 copy of the track in orange or 2 copies in green really correlated nicely with the length of the repeat in each of these individuals as estimated by some of the short read data in some of these 1,000 Genomes Project samples. What's surprising is that actually some of the Denisovan and Neanderthal alleles were not expanded the way that they were in American populations, with the caveat that there's really only one Denisovan allele that's been that has been sequenced at this point. And I'll refer you to this preprint for more details. As kind of an independent confirmation that this repeat is associated with the signatures of positive selection, there's been other groups that have identified genome-wide SNPs that are associated with positive selection in North American populations. They also came across MUC19 and had a couple of SNPs that are their key candidates as associated with this positive selection. And if we take a look at some of the variants in their study, we could also see that, especially in American populations, the alternative allele tagged larger numbers of repeat copies, again indicating that this repeat expansion has led to some kind of signatures of positive selection in individuals. We did see individuals in South American samples that had this variant, but they did not have this expansion. So, okay. So again, we always have these questions that, this is all great. We find these repeat expansions, but what is the function of this? And because this is a 12 kb exon that jumps to 25, almost 25 kb in length, it's very difficult to model using, cell lines or model organisms. So we really wanted to kind of use alternative approaches to see why this repeat had expanded to such a degree in American populations. And so for this, we took advantage of some of the kind of increasing numbers of biobank data that's become available, where individuals have donated their DNA for whole genome sequencing and coupled these often with electronic health records. And some of these studies have been done throughout the world. And this NIH All of Us data set has really kind of exploded in the United States to try to sequence up to 1 million individuals and combine this with electronic health records. And this is really to help address some of the health disparities and disparities in some of the genome-wide association studies that have been done up until about 5 years ago that were primarily of individuals of European ancestry, much more disproportionate than the global population of individuals. And this is work published by Alicia Martin. And so what the All of Us research program that we've been starting to kind of work with extensively is has done is recruited individuals from historically underrepresented groups, so both from different from races that are different, historically underrepresented, but as well as income, age, sexual and gender minorities, and education. There's a lot more individuals of rural ancestry, older individuals, and groups that have not been included in as much in previous biobanks. And there are a considerable number of surveys that have also been individuals have responded to help address, whether there are differences in health disparities that could impact some of the disease associations and genetic variants that are identified. All of this work from the NIH All of Us is done on a cloud environment. And so we had a bioinformatician, Evangelos Nizemis, who was able to kind of assess this and figure out all the nuances of some of the data analysis to perform what we really wanted to do, which was to identify whether there were any particular phenotypes associated with this MUC19 repeat expansion. So to do this, we took advantage of Josh Denny's PheWAS approach, and this is kind of turning a GWAS on its head. So in genome-wide association studies, you have a particular condition, and you're looking for which genetic variants are significantly associated with that phenotype of interest. In PheWASs, you actually are starting with a particular genetic variant, and then trying to identify whether certain phenotypes, like skin cancer here, are enriched or depleted as a function of individuals that have this genetic variant. And so there are our PheWAS pipelines that are available, and I'm happy to kind of go over some of these details if people are interested offline. But with these pipelines, you can replicate, fairly well-established traits associated with particular genetic variants. So if you throw RS334 into the PheWAS pipeline, sickle cell anemia comes up with a negative log 10 value of 110. And an odds ratio of over 700. So this is a very, very well-established variant associated with sickle cell anemia. There's a deletion in the CFTR gene, del508, that's fairly well associated with cystic fibrosis. And again, you can see the p-values are negative log 10 p-values are 160 or more with really high odds ratios for this variant. So we could replicate existing findings. We could replicate the ApoE4 variant that is a strong risk factor for Alzheimer's disease. You can see that, if you introduce this RS429358, which is the defining variant of ApoE4, you have increased risk of Alzheimer's disease, memory loss, and mild cognitive impairment, as well as a lot of lipid traits that are also associated with ApoE, which is apolipoprotein biology. And then conversely, a protective variant, ApoE2, is associated with protection against the same lipid trait. So if it's a risk, it's an upward triangle. If it's protection, it's downward triangle. And we saw protection against Alzheimer's and dementia-related traits as well. And this is kind of what the forest plot looks like, again, where you can see these elevated odds ratios for Alzheimer's disease associated with the ApoE4 allele. So we were interested not only to do a PheWAS, but also kind of, as proof of principle, can we use some of this information, like I was alluding to before, at estimating repeat length using short read data uh, at the MUC19 locus. And so again, for this, we would count the number of reads across the VNTR and compare it to the average number of reads at non-repetitive regions. And at MUC19, this is downsampled to 1 every 10 individuals. You could see that the reference allele has 287 copies. For the MUC19 repeat expansion, and any individual that had up to doubling of that repeat length was predicted to be American ancestry. And similarly, the selective variant was also associated, the positively selected variant was also associated with either 1 or 2 copies of the 1 or 2 copies of the protective variant was associated with expansion and doubling of the MUC19 VNTR. So PheWAS of this VNTR and that surrogate SNP that I had in the previous slide didn't reveal anything that was significant after Bonferroni correction for 1800 phenotypic traits. However, there still are some interesting kind of examples of conditions that seem to be protected um, in expansion carriers. So again, this is at 33,000 individuals of Hispanic ancestry. We actually used principal component analysis and correcting for age, sex, the first 3 or 8 PCs to identify phenotypes associated with individuals that were carrying this repeat expansion. And some of the top protected traits or conditions like major puerperal infection. I had to look up what this actually means, but these are severe infections after birth, so in mothers. And so if this repeat expansion is protecting against some of these infections after birth, perhaps modern medicine has been able to promote survival of individuals that, otherwise would succumb to this. But, 500,000 years ago, this might have been a much different story, and there may have individuals that did not have this repeat expansion may have died off, and it could explain why this repeat expansion is in such high abundance in American samples. And we could also use the information in a very similar vein to identify other repeats in other mucin genes. So this is work that Lizzie Plender and Evan Eichler's group had done to identify patterns of VNTR expansions in other mucin, MUC5A and MUC5B. And you can see again that these coding exons cover most of this, the repeat expansions cover most of the coding exon. And you can do PheWAS analyses of some of these variants to identify variants that are associated with things like lung disorders like fibrosis. So in summary, I'd like to kind of conclude by saying that, like some of the repeat expansions that we started with that are risk factors for neurodegenerative disease, perhaps have had some type of protective features throughout human evolution, and that not all repeat expansions are bad. The one that we identified in MUC19 has a protection against both puerperal infection as well as other viral insults. And this could be consistent with this idea that the mucus barrier, which is normally fairly thick, can be degraded by pathogens. This repeat expansion that doubles the size of the mucus barrier could act as like kind of a castle wall, a doubling of the front line of defense against some of these pathogens in various tissues. And again, could explain the signature of positive selection on this and on the Denisovan on the Denisovan haplotype. So with that, I'd like to thank our funding sources, members of the lab, especially Eli and Evangelos, that were doing a lot of the computational work. Meredith, of course, who has gone on to her own group at Colorado College, had done a lot of work on the WDR7 repeat expansion, and this is kind of a repeat expansion scarf she got right before leaving for her own position. So with that, thank you for your attention. I'm happy to take any questions.
Cool. That was really great. Thank you, Paul. It was really interesting. I had actually never realized that there were any of these expansions that were actually protective. So that's interesting to see, and interesting to see that mechanism of how it would work. One question I had is I kind of always had the impression that these sort of ratcheted in one direction, always getting bigger. But you did, and it seemed like that was mostly the case with all of your plots, but you did see some deletions, so it looks like it can collapse. So, what do you think's the dynamic there?
Yeah, exactly. I mean, the more we start to read into some of these repeats, the more we're starting to kind of see some of the patterns. And um, a lot of the signatures we seem to see at the beginning and end of the repeat motifs is just slight differences in the length. And so if the repeat is 25 base pairs, the first couple of repeats might be 27 or 28 base pairs, and the last one might be, also different in length or fairly variable in sequence composition. And then a lot of the variation, for the most part, it does seem to be happening in the middle of the repeat itself. Some of the expansions and contractions. The first example that I provided with WDR7, where there was this directionality in the expansion, seemed to be more the exception rather than the rule.
Hmm.
We thought that this was going to be, more universally kind of attributed to other VNTRs, but it seems like a lot of the variations seem to be happening internally at the repeat.
Do you think any of the directionality could be sort of position related, sort of like whether it's on the leading or lagging strand when there's replication
Yeah.
happening?
Yeah, we've tried to test that with folks in genome sciences to try to see, is there, like a replication of origin that is determining whether there is expansion that's occurring depending on where the proximity to leading and lagging strand, like you mentioned. Um, and so we don't have good evidence for that
Hmm.
yet, but that's certainly something that we're interested in kind of pursuing is, like, yeah, how that could impact both the expansion events and the directionality.
Okay, that's interesting. That's definitely room for other questions. If anybody has them, they can put them in the chat or raise their hand. Um, yeah, I think these little repeats are really fascinating. I still remember it was actually one of my first introductions to the idea that of sort of survivor effects and mutations is because when I first started hearing about these, I was wondering why are they always occurring in threes? Why is it
Hmm.
always three base pair expansions? Um, and why couldn't there be two or four in and the answer that I heard at the time, which I think was when I was at PA at Barbara Wakamoto's class,
Hmm.
uh, was that if you have anything other than a three base pair repeat expansion in the middle of an ORF, it just kills it, and then that's probably lethal, and you don't see it. But these three base
Hmm.
pair repeat ones end up actually sort of, in many cases, they end up being not lethal, and like you've seen, sometimes protective, but then sometimes just causing sort of more subtle phenotypic variation depending on the sequence and the expansion. And so it actually makes for a much more sort of plastic change to the behavior, which is interesting.
Yeah, absolutely. And I think one of the fascinating questions that we have for some of these repeat expansion disorders is why there is certain tissue specificity as well. So why, the CAG repeat expansion in Huntington leads only to striatal degeneration in Huntington's disease versus hexanucleotide repeat in C9orf72 is going to lead to degeneration of uh, uh, and particular expansion in frontal cortex. And then you can have pentanucleotide repeats
Hmm.
in some of the spinal cerebral ataxia genes. And so this five base pair repeat motif leads to cerebellar degeneration. And so what are the cofactors or features that enable some of these somatic expansions and tissue specific phenotypic effects is still a really kind of open question in the field.
Yeah, we got a question
Yeah.
from Rick.
Okay.
Great talk, Paul. Can you show us some more details about how the phenotype loss works, the Phi loss? How much diversity and sequence do you need? What kinds of phenotypes are likely to arise as
Yeah,
significant?
it's significant. Yeah. Okay. So the FIWAS, again, it's a kind of a computational pipeline where you will introduce, your variant of interest, whether there's zero, one, or two copies um, of that variant in individuals. And then it'll actually iteratively go through, the 1800 phenotypes and count, individuals that have, that are homozygous for the minor allele of, a particular variant, how likely are they to develop fibrosis or some other condition? Um, and so I think that the idea is that there aren't necessarily a priori any phenotypes that should show up as significant, more likely than others, unless there is a lot more genetic heterogeneity in some of these disorders. Um, so, for instance, for some of the repeat expansions that we've evaluated, we haven't really seen too many like cancers showing up as phenotypes that are picking up. Um, That said, I mean, there are certainly, there's a lot of underlying kind of work that we need to do to try to um, adjust for some of the variables in the analysis, especially in the or in the collection samples from all of us. So, for instance, samples are, the amount of samples that are being collected is certainly improving, but there are like certain regional areas that have a high number of cases. So there's a lot of, for instance, Hispanic samples, both from New York City as well as Arizona. And you can imagine that individuals in Arizona versus New York City are going to have quite a bit of different phenotypes that are associated with more just urban versus rural living and geographic proximity. So correcting out and adjusting out some of those variables is really important in some of this analysis to avoid kind of issues just with population structure.
Okay, great. We're getting a couple more. There's a question from Balawi. Do you know of any limitations in capturing clinical irrelevant repeats or known repeats in general using long read technologies versus short read ones, i.e., cases where actually long read technologies would miss those repeats?
I, I I think one of the limitations is the cost associated with whole genome long read sequencing. And so if you have a rare case, trying to be able to identify those and capture them, it can be challenging, simply because there isn't a very good, nomad database for, which repeats are actually causal for disease. So if you're looking for a Mendelian condition, it may be that there's you identify an expansion, but it might not be disease causal. Um, and maybe just like at low frequency in the population. There are other issues where, there can be very large structural variation or areas of like, again, in the telomeres or centromeric regions where, resolving the exact composition of some of these repeats can involve requiring high depth of coverage. But a lot of the limitations from short read have sequencing technology have resolved. So we can capture things that have high GC content, for instance, or other situations that previously were intractable to sequencing.
Good.
It certainly won't capture everything, but yeah.
Looks like Mark Reeder has a follow-up on Rick's question saying,
Mm.
since some of those repeat expansions are population specific, yeah, do you correct for it? I guess, how do you correct for that in the PWAS studies?
Yeah, and so we do multiple different strategies to be able to try to correct for population structure. First would be to add various principal components into the model and adjust for those, and that does get rid of a lot of the variability in population structure. And another thing that we can do is, again, a lot of these individuals from the All of Us data set already have their global ancestry compositions based on principal component
Mm,
calculations. And so you can actually then just bin these individuals and look, say, for instance, only in American populations, only in individuals of African ancestry, what the uh, what the PheWAS results will provide. So we do global analysis, but then we also do population specific analysis, and then even we'll dive even further, and look at, for instance, local ancestry. So some of the calculations that we need to do at ApoE that has a lot of differences by a population of origin. We won't even consider we won't consider as much the global ancestry, but the local ancestry at that site itself to identify and classify groups based on the local ancestry at ApoE for what phenotypes may be associated.
great. I guess I have one other quick one that just I thought of. Sorry to ask so many questions.
Right.
Um, you talk a lot about the genotypes of the individuals as a sort of a single genotype, but is there somatic expansion of these within tissues, and does that lead to disease in any of these cases? And sort of how would you try to
How would you
look at
parse
that?
that out?
Yeah.
Yeah, and that's where again, yeah, so again going back to kind of patient specific cohorts and databases and looking at, you know, differences in long, for instance, long read sequencing in the brain versus the blood can help indicate, where some of that somatic instability is going to be taking place. And there certainly are risk factors that can contribute to some of the somatic instability. So individuals, for instance, in Huntington's disease, um, if they have variants in some of the mismatch repair genes like MSH3, those variants are going to then predispose individuals to more somatic instability or less, and that's going to impact, again, phenotype, age of onset of some of the phenotypes there. So it's certainly not addressed in some of the models where we're looking only on blood, but adding in other tissues could help assess what some of these somatic variants or somatic instability component to that calculation.
Cool. That's really interesting. Okay. I think if anyone has any last minute questions, they can ask it now. But otherwise, I want to thank everyone for coming to the talk, and definitely give an extra thanks to Paul Valdmanis. This has been really awesome to hear about, and definitely got me thinking about a lot of other things that haven't, a lot of types of mutations that I don't think of usually, and thinking about them in new ways and new consequences and things. Oh yes, we have our next in the series with Bill Hagopian on July 9th. There's the link that's in the chat here. And that just popped up. All right. Yeah, thank you very much. It's great to talk to you, and see you.
Super. Thank you.